So to preface my talk, as one of many people here developing and training AI models, I really love data. However, this data, especially for people in biological fields, can be very expensive. So my talk will be about how we can acquire the low quality but less expensive data and use AI to increase the quality of the data after the fact. So the data I'm working with is known as gene expression data. And to give a very quick summary, the cells in our body contain DNA, which contain genes, which produce RNA, which produce proteins that do most of the work in our body. All this is not all that important. What is important, though, is that the DNA, which contains genes, doesn't really change. Between us a little bit, but inside of us doesn't really at all. But what is interesting is that depending on the state of the cell, that is to say, what type of cell it is, what is it doing, is it cancerous, is it healthy, the amount of RNA each gene will produce will vary. Thus, if we want a snapshot into what is happening inside of cells, say inside of a tumor, we can take some of its RNA, sequence it, which allows us to essentially get an approximate count of the amount of RNA produced by each of the genes. In our case, about 20,000 genes. Let's say we rinse and repeat that for several thousand cancer patients. We can see that we're starting to build up a very rich data set for all sorts of machine learning purposes in cancer research. And this is just specifically cancer, it can be anything. But the quality of the data will depend on the amount of RNA we're sequencing, which is known as sequencing depth. And unfortunately, at the standard amounts of sequencing, just for the sequencing costs, it's around $100 per sample, which is a lot and can be reasonable in smaller experiments, but we want to train models, and models require tens of thousands to millions of samples, meaning that this starts to become very pricey. Naturally, if we reduce the amount of sequencing, we can also reduce the costs, but this comes at compromising the quality of the data. So how can we keep the data cheap, but still get it good? And our first instinct was to train a neural network that will take the low-depth RNA-seq data and predict what it would have looked like had we sequenced at the normal levels. And for the most part, it works well. We're able to recover a lot of the correlation. We're able to recover a lot of lost information in terms of the downstream analyses. But what we notice is that as we reduce the sequencing depth, more and more, the model will just give us the average sample of everything it's seen in the training set. And that makes sense. The amount of data, the low-depth data seems to look like itself a lot more. And thus, there's more variety. But now the model isn't telling us when it's not sure what the real answer is anymore. So we need a way to essentially tell us when it's too uncertain to pinpoint exactly one sample what it would look like. And so for that, we are instead moving to a more probabilistic approach using Bayesian inference. I won't go into much detail about that, so don't need to know too much. It's just that we're combining two probability distributions. One, which encodes our prior beliefs, which is surprisingly complicated. We need to model RNA-seq data, which is multimodal, which has a lot of correlations, which has approximate distributions on genes. It's complicated. So we used a neural network called a normalizing flow, which converts these complicated data into an easy distribution. Afterwards, we also used the likelihood of the observed data, which is very easy. It's just a multinomial distribution. Combine that with Bayesian sampling. All that is details. What is really interesting is that this allows us to instead get a variety of plausible samples instead of just one. And the more uncertain the model is, the wider the breadth of the plausible samples will be. It's currently in progress, and the main weakness is that it's slower than a neural network, but it might be able to encode more properly the kind of data we want to get when recovering the sequencing depth. So To conclude, there are many types of data that are expensive. Here I presented 2 ways we can recover quality while using data cheaply. And obviously, this will lead us to get data sets for much larger data sets for a smaller cost. But I want to leave everyone with a little practical tip. Do check that your models are not just predicting an average. It's harder to check than you think. Thank you very much.